Thank you very much, Nikolai. That was extremely generous. I have to say, it is intimidating speaking in front of close colleagues and friends who know much more about this probably than I do, and about things that they've heard about before. But I'm very appreciative of the organizers for allowing alcohol to be served before the talk began. I thought today that I'd organize this around obviously targeted protein degradation, but how the field has, every single time we've embarked on a project, has ended up somewhere very unexpected. And that's been a hallmark in the field, that people have said that there's a lot of serendipity about the molecules that are discovered, but in many ways that's just the way science works, that we identify new mechanisms, particularly with regard to protein-protein interactions, which are so challenging to manipulate, but very frequently end up completely not where we expected. These are my disclosures. Neomorph is a company that is interested in targeted protein degradation and funding the beer bill. So I'll start where this all began for us and for many in the field. So thalidomide, as you all know, this is a well-trodden story, but it was first used in the late 1950s as a medication that had antiemetic properties, sedative properties, and was thought to be totally safe because of its, in part at least, because of its lack of toxicity in animal models in preclinical studies in mice and in rats. And in 1960, a young pharmacologist, Frances Kelsey, was hired at the FDA. So a few years after the drug started to be used, she was one of only 7 full-time physicians at the FDA approving drugs. And in those days, the companies could just call up her boss and say, come on, do this as quickly as you can so we can start having the drug approved and distributed. And she said that she didn't see enough safety data, particularly in pregnancy, to allow the drug to be approved. And sure enough, within months or a year after she blocked it, the first reports of phocomelia, these limb bud defects, were reported. It actually took quite some time to put 2 and 2 together because any individual physician might only see one instance of this, and even for rare things, it is hard to associate a rare condition with a drug that was thought to be so safe and was even over the counter in so many places, approved in over 20 countries. But she blocked it from being approved in the United States. And just in 1962, just 2 years after she started the FDA, a year before Kennedy was assassinated, he gave this award, Distinguished Civil Service Award, to Frances Kelsey. So this is the beginning of so many stories. It's the beginning of a transformation at the FDA. Frances Kelsey actually worked there till 2005 and was behind many of the changes at the FDA that now everybody goes through to get their drugs approved. Certainly an enormous event in the field of teratogenicity and how to prevent it in the future. It was the beginning of blockbuster drugs, thalidomide, lenalidomide, and pomalidomide, before any understanding of their mechanism of action, and hopefully, most importantly, giving birth to the field of targeted protein degradation, which we all are very much part of. So, as you all know, the thalidomide was derivatized with relatively minor modifications, but far increased the potency of the molecules in some cellular assays and led to just tremendous efficacy against a number of targets. And I think this is also a tribute to good clinical research. The drugs were tested widely in cancer without really understanding where there would be efficacy. It was tested, thalidomide was tested in multiple myeloma, at least with a rationale of inhibiting angiogenesis and that ultimately didn't have anything to do with the clinical activity, but it worked. The clinical trials were positive, leading to FDA approval. In myelodysplastic syndrome, a phase 1 trial, again, without a clear rationale other than angiogenesis for trying thalidomide or lenalidomide and MDS, and about 5 patients in that first trial happened to have deletions in chromosome 5q, and they had complete responses, which we almost never see in MDS, and that led to FDA approval after a small phase 2 trial that was not even randomized. So these drugs have phenomenal clinical activity, and most of that clinical development really happened just from clinical trials and have led to massive changes in the treatment of myeloma in particular and lymphoid malignancies and standard of care for del(5q) MDS. So with that activity, I happened to work on del(5q) MDS since the very beginning of my postdoc, and in fact, halfway through that postdoc, one of our leukemia attendings, Rich Stone, said, "Why are you doing that? It's solved already. There's no reason to work on that one anymore because we know del(5q) can be cured." But it stayed working in that field and really wanting to understand how these drugs worked. And Hiroshi Honda's lab made a derivative of thalidomide that they pulled down, identified this Cullen ring ligase as a direct target, and cereblon is the potential direct target of thalidomide. We did a similar experiment with lenalidomide, derivatizing it, pulling down the same complex. And both the Honda lab and our initial studies completely assumed, as one would expect, that these are small molecules that inhibit this enzyme. Almost all molecules that bind an enzyme are inhibitors of that complex. We spent years looking at auto-ubiquitination of cereblon and inhibition of that by lenalidomide, which it actually does, looking at theoretical other substrates, all of which turned out to be negative, that yielded nothing until finally we did a proteomics experiment with Steve Carr's group at the Broad Institute. And it was sufficiently surprising that I was 100% sure that my postdoc had swapped the tubes, because we were 100% expecting and only looking for proteins that would accumulate after treatment with lenalidomide. If you inhibit a ubiquitin ligase that targets protein for destruction, you would look for proteins that accumulate. We actually, this was like the third go. There was always something wrong with the experiments. The amino acid concentration, this was a SILAC experiment. One of the labeled amino acids would be at slightly wrong concentration. The cells would grow at different concentrations, skew the proteome. Finally, we got a clean result, and it was backwards. So I thought, oh my gosh, here we go again. But he did do Western blots and convinced us all that these proteins, IKZF1 and IKZF3, otherwise known as Ikaros and IL-2, are depleted very rapidly in a dose-dependent way after treatment with lenalidomide. So something we didn't expect, and were far out of our depths when this finding was made. So I think, as most of you know, then lenalidomide, thalidomide, pomalidomide bind CRBN. When the drug is bound, they have increased affinity for IKZF1 and IKZF3. And when the proteins are brought into proximity with a Z3 ubiquitin ligase, it's polyubiquitinated and degraded. IKZF1 and 3 are well-studied master transcription factors that are essential for multiple steps in lymphoid differentiation, and they also happen to be essential for the survival of multiple myeloma cells, and their depletion leads to killing of multiple myeloma cells. So that provided a mechanism of action for the activity of the drugs in multiple myeloma. It also provided a mechanism for the induction of interleukin-2, one of the other canonical activities of lenalidomide, and interleukin-2 is repressed by IKZF1 and 3, and so the depletion leads to transcriptional release and expression of interleukin-2. At that stage in the game, I have to admit, I wasn't quite sure we had the whole story. These approaches of making a derivative of these molecules and pulling them down and getting these ubiquitin ligases, they were kind of clean experiments, but they weren't perfect, and definitely wouldn't have bet the farm that we had the whole story, that there might be other ubiquitin ligases or other proteins that are targeted by these molecules. And this is the experiment that sort of convinced me that maybe this is really the only game in town. So this was a genome-wide CRISPR screen that Quinn Sievers initially did of looking for resistance to lenalidomide in multiple myeloma cells and M1S cells. And he's a super careful scientist, really worked out the screening conditions elaborately and beautifully, and did this screen. This was one of our first, maybe our first genome-wide CRISPR screen that we did in the lab. And out of 6 guides per gene, 20,000 genes, hits 1, 2, 3, 4, 5, 6 were all cereblon by a long shot, nothing close. And out of the top 20 or 30 hits, we got almost every component of the E3 ligase system, the CRL4, CRBN, E3 ligase, including the E2 enzymes, the entire netylation cycle, everything that was essential for this. One of the reasons that this worked so well and has continued to work so well is this is an extremely unusual situation for genetic screens. You've got, normally, if you're thinking about a drug that inhibits an enzyme, if you target that enzyme, the cell may die if it's an anti-cancer drug. Here, we have a drug that needs this ubiquitin ligase to act as a molecular glue to target its substrates. And if you deplete any aspect of that enzymatic complex that's essential for the drug activity, you get resistance to the drug before the cell dies, even for the components that are cell essential. We've repeated this more recently with molecules that target GSPT1 for degradation. I should say these screens work very well, both if you do the screen for with degradation as a readout or resistance as a readout. This is a resistance screen for GSPT1, or actually, sorry, this was a degradation screen for GSPT1, and it was identical results. So the entire complex is the same E2 enzymes, the same netylation components are identical. So these different molecules that target different proteins, have different mechanisms of cell killing, require the same components. And again, these are just beautifully clean systems for doing genetic screens because of the requirement of all of these enzymes for the activity of the drug. So that was the first highly unexpected direction where we expected to be studying inhibitors of an E3 ligase and wound up with these molecular glues. The second, which maybe should have been apparent to us from the beginning but wasn't, was the story of whether these drugs work in murine models, and with some recent updates to that. So this part began with trying to understand our original goal in the field, which was to understand why lenalidomide works in del(5q) MDS. Further proteomics revealed another substrate, casein kinase 1 alpha, and that immediately caught our eye because it's one of only 40 genes in the common deleted region for del(5q) MDS. And del(5q) MDS is very consistent and unusual in that the deletions are universally heterozygous. They're never homozygous deletions in chromosome 5q, and there's a number of genes in there that are essential, so cells would never tolerate homozygous deletions. So haploinsufficiency for several genes drives the phenotype of del(5q) MDS, and CK1alpha is one of those genes. And even within genetic deletions in cancer where one allele is deleted, most of the genes are expressed at normal levels because there's homeostatic mechanisms for recovering normal gene expression. CK1alpha is one of the few genes in del(5q) that have true haploinsufficient 50% mRNA and protein expression. So since CK1alpha is an essential gene, always cells die if they lose all of CK1alpha, that gave us the hypothesis that if cells have 50% expression and CK1alpha is degraded further, they'll have a therapeutic window relative to cells with the full CK1alpha expression. And this was clearly a bona fide substrate. We could validate that there's an interaction between CK1alpha and CRBN that depends on lenalidomide, that ubiquitination of CK1alpha is dependent on lenalidomide. Eric Fisher and others identified a nicotome, identified a crystal structure ultimately of this interaction. So it's a bona fide substrate. But to really understand in vivo how it was working, we needed to make a CK1alpha heterozygous knockout mouse limited to the hematopoietic system. We generated this mouse, characterized it. It has a lot of interesting properties as a gene, regulating beta-catenin as well as p53. But when we treated with lenalidomide, we had no effect. And we did, it was a crazy experiment over Christmas. The postdoc saw no effect. She massively increased the doses in frustration, still absolutely no effect. And we thought we might have our whole hypothesis wrong until she did a cell line experiment, BAF3 cells, a leukemia cell line that we use all the time, and treating with lenalidomide resulted in absolutely no change in CK1alpha expression. And then ultimately realizing that we couldn't find any evidence in the literature that these compounds had clinical activity or toxicity in murine models, and that maybe we had a mouse-human defect. Ultimately, we were able to show that these mouse cells had no response to lenalidomide, but if we identified a single amino acid change, probably the most conservative one in the gene, that if we change that one amino acid, the isoleucine to valine at position 391, that we could get and express that cDNA in a murine cell, we got nice degradation. So that one mutation can prevent the activity of thalidomide, lenalidomide, pomalidomide in mouse cells. We made a germline knock-in of that single amino acid, so not a conditional, just changing one amino acid in the germline of the mouse. Mice are completely fine. We find no defects in them at all. Hematopoietic system's normal. And if we do whole proteome experiments and look at what proteins are degraded, in wild-type c-KIT positive hematopoietic stem progenitor cells, we see nothing degraded at all. Homozygous knock-in mice, c-KIT positive cells degrade CK1alpha and IKZF1 as expected. Treating with pomalidomide degrades IKZF1 and other zinc finger proteins, but not CK1alpha, which is consistent with the lack of activity of pomalidomide in del(5q) MDS. So recapitulating what we expect in humans in a mouse model, enabling us to finally do in vivo experiments in mouse cells. This change lines up very nicely with teratogenicity, where organisms like humans and rabbits that have thalidomide teratogenicity have the human residue, whereas the mouse residue is present in organisms that do not have thalidomide teratogenicity. And this particular amino acid is actually not in the drug binding pocket and is actually adjacent. It's right where substrates bind, including IKZF1, 3, and CK1alpha, preventing their binding, but it actually does not impair binding of lenalidomide or thalidomide into that pocket, which is why PROTACs work. And actually, I remember Jay emailing as they were publishing their first degradation paper showing that that you can make these beautiful PROTACs based on thalidomide derivatives, and said, wait a sec, we're getting activity in mice. Is this, do you have something wrong? Do we have something wrong? But that makes perfect sense. The PROTACs will bind into that pocket just as well as thalidomide or lenalidomide. The substrates that require that proximal amino acid for the protein-protein interaction do not get degraded, but these neo-substrates are prevented. So when we look at pregnant mice, the main thing that we saw when we treated mice that had the double homozygous knock-in with lenalidomide or pomalidomide was loss of pregnancy. And in fact, that may be the most common effect of the drugs in humans. This was a drug used in the first trimester, and miscarriages are common, and weren't being tracked in any way, but there are estimates that as many as 100,000 pregnancies were lost due to thalidomide, and that's very much what we see here, that only in the knock-in, only in mice treated with lenalidomide do we see this pregnancy loss. Now, we didn't see limba defects, but Eric's lab identified, using this mass spec experiment in embryonic stem cells, that SALL4 is a protein that is expressed in embryogenesis, not expressed in many adult tissues, is powerfully degraded by thalidomide as well as other thalidomide derivatives, and amazingly, SALL4 is mutated in the germline in a syndrome that phenocopies the lenalidomide, the thalidomide toxicity beautifully. So the limba defects, other cardiac defects, ear defects, et cetera, that are seen with thalidomide are phenocopied perfectly in this germline syndrome, and to the extent that there were individual cases of this germline mutation being misdiagnosed as a case of thalidomide teratogenicity. So this appears to be the critical gene that is degraded during embryogenesis that mediates thalidomide teratogenicity. Now, we thought we had this story all done and dusted, but started working on small molecules that induce degradation of a protein called GSPT1, which including CC90009, CC885. These are molecules identified and developed at Celgene. CC90009 is in clinical trials for leukemia right now. We wanted to see if we could, as a leukemia-oriented lab, see the activity of these molecules, but we did not see activity of GSPT1 degradation in the mouse models we've made. So you can see here that in the single knock-in mice, when we treat with a GSPT1 degrader, we get no degradation of GSPT1, but we identified another amino acid at position 380 that if you mutate that mouse residue to the human residue and express that in a mouse cell, you get nice degradation. So there's a second residue required for GSPT1 degradation that is not required for Ikaros, IL-2, CK1alpha degradation. We've made leukemia cells expressing these various mutations in CRBN. You can see that wild-type mouse cells are not killed by CC90009, but any cell that expresses a version of cereblon that mutates both the I391V and the V380 residues are nicely killed by CC90009. So we've now made a second mouse, a double knock-in mouse with the V380E and the I391V. These degrade GSPT1 very nicely, whereas the single knock-in mice do not degrade GSPT1 at all. We've made leukemias from this double knock-in mice, and you can see that leukemias from the double knock-in are killed very well by a GSPT1 degrader, whereas wild-type MLAF9 leukemias are not degraded or not killed at all. So we hope that this is now the end of the story, that these are the only two amino acids that are required for the activity of cereblon targeting degraders to induce degradation in murine cells. And now we have a tool to test these molecules in vivo. And this mouse is now at Jackson Labs and hopefully will be available very soon for anybody who needs it. So to validate this at a proteome-wide scale, we did whole cell proteomics with Eric. We could see that in wild-type mice treated with CC90009, we didn't see any degradation in the cell line. The single knock-in mice, nothing degraded, but in the double knock-in mice, homozygous double knock-in mice, GSPT1 and GSPT2 are both nicely degraded. So now we can do some in vivo experiments. An example is understanding the role of GSPT1 degradation in normal hematopoiesis. These are competitive transplant experiments where 50% of the hematopoietic cells have the double knock-in, 50% of the cells have wild-type cells. We can treat those mice with a couple pulses of CC90009, and you can see that it's a very toxic compound. GSPT1 is a protein that's critical for protein translation, in particular for translation termination, which is important for all cells. And you can see that the chimerism falls in mice in the cells that express the double mutant, and in B cells, T cells, myeloid cells, and progenitor cells, you can see a marked decrease in chimerism from the double knock-in cells being killed. However, long-term hematopoietic stem cells, which are more quiescent and therefore probably require less new protein translation, are relatively preserved when exposed to CC90009, which potentially can give a therapeutic window given that the leukemia cells are killed so potently by CC90009. And we checked that these long-term hematopoietic stem cells are not just, the number isn't just decreased, but their function is preserved. We took those long-term hematopoietic stem cells, transplanted them into a secondary mouse, and followed them for about 4 months and showed that functionally, those remaining long-term hematopoietic stem cells are just fine, and the number of long-term hematopoietic stem cells at the end of that experiment is preserved. So at least potentially, this drug could be used to treat leukemia with preservation of long-term hematopoietic stem cells, killing of the progenitor cells, and killing of the leukemia cells. Thinking about, I didn't include it, one of the potential therapeutic indications for this is p53 mutant leukemias, which is the worst of the worst, almost universally fatal type of leukemia with resistance to most of the drugs that we use, including anthracyclines and Ara-C. And whenever we do these in vitro or in vivo studies with Ara-C and a p53 mutant leukemia, the p53 mutant leukemia cells are resistant to the treatments. When we treated with CC90009, the p53 mutant cells were equally sensitive as non-p53 mutant cells to GSPT1 degradation. So at least a possibility that that might be a form of leukemia that would warrant testing with GSPT1 degraders. All right, so as we've tried to expand the number of E3 ligases or mechanisms of targeted protein degradation, every story has led in a new direction, and that's continued in the lab with stories that are just halfway through right now, but I think these stories illustrate that there are many, many mechanisms of induced proximity and targeted protein degradation to be found that hopefully some will lead to new therapeutic opportunities. So this first one was led by Mikolai and colleagues, and Mikolai had the creative idea, as Roddick said, if you ever want to brainstorm with somebody, there's nobody better in the world to do that with than Mikolai. And one of the approaches that has been used at the Broad Institute is to look for correlations between the level of expression of a gene and the activity of a small molecule. And while that seems incredible that that could work, it has consistently identified the direct targets of therapeutic agents. And so he looked very broadly for small molecules that would have activity that correlates with the expression of any E3 ligase component. And he did this with a large data set from PRISM, which is a multiplexed assay across many cell lines that has IC50s for about 4,000 compounds at the time that he did this, across about 500 cell lines. So in that data set, can we find small molecules that kill cells in a way that correlates with the expression of an E3 ligase component? He had about 96 hits, which correlated with the number of wells in a plate. About 95 of them or 94 of them did not validate, but one of them, this molecule CR8, which had activity that correlates with the expression of a protein DDB1, validated in his initial studies. And here's a validation assay. In black, the killing of cells with CR8, which is a CDK inhibitor, and then in red with knockdown of DDB1. DDB1 is an essential protein. Normally knockdown of DDB1 kills cells, but here it actually rescues cells from killing by CR8. So that's, it's not just a toxic shRNA that sensitizes, but actually a toxic shRNA that rescues cells from cytotoxicity from CR8. So DDB1 happens to be an adapter protein that is used by the CRL4-CRBN system that we talked about with thalidomide. And CR8 seems to do something to this E3 ligase system that we are eager to understand. So first, needed to understand what protein might be degraded. So did a proteomics experiment where he looked for what proteins might be rapidly degraded following treatment with CR8, and found that Cyclin K was rapidly degraded by CR8. And by Western blot, you can see it almost completely go away. And if you treat with an E1 inhibitor, an etylation inhibitor, or proteasome inhibitor, you completely rescue the degradation. So targeted protein degradation. I have to say at this stage, I completely didn't believe it. CR8 is an inhibitor of CDK12, which is a binding partner of Cyclin K. I thought we just had some toxic mechanism that somewhere in the pathway had an E3 ligase involved that was important for the cytotoxicity, but thought we had a long way to go to show that there might be a direct effect. But Mikolai is not only very creative, but very persistent and kept hammering away at this. He did a beautiful genome-wide CRISPR screen looking for resistance to CR8, and elegantly, DDB1 scored at the very top. So knockdown of DDB1, the original hit in his informatics screen, rescued CR8. He also found RBX1 and COL4B, which are part of this E3 ligase system. So now we're starting to really put some of the pictures together. DDB1, COL4B, RBX1, Cyclin K is the protein getting degraded, but the all-important substrate receptor was missing. And in the case of thalidomide analogs, again, this would be cereblon, and that is where the molecular glue interaction occurs between the substrate and the substrate receptor. So we needed to try to find that. His next screen was a degradation screen. So what are the proteins required, or what are the genes required for degradation of Cyclin K? He did this screen, and again, DDB1 scored at the top, which is very reassuring. He did not find any substrate receptor, which was starting to be frustrating. He did find CDK12, and that's a little bit unusual in that CDK12, which is the direct target, which we know is inhibited by CR8, is important for the degradation of Cyclin K. And so that gave the hypothesis that maybe CDK12 is actually part of the complex that is required for Cyclin K degradation. The breakthrough in this then came from Nico Tomei's lab and Georg Petzold, who did an in vitro ubiquitination assay and only put into the assay recombinant CDK12, CR8, the small molecule, DDB1, COL4B, and RBX1, and the E2s, and no other proteins. So we thought we might be missing a substrate receptor, but he did not add any substrate receptor in. And with just these components, he could see this ubiquitination laddering, indicating that we had every component necessary for ubiquitination in this in vitro recombinant assay. And then they managed to get a crystal structure of DDB1, CR8, CDK12, and Cyclin K. And you can see that CR8 is indeed a molecular glue, but it's not at all what we expected. We expected a glue between a substrate receptor and a substrate, but instead we had a glue between DDB1, an adapter protein, and CDK12, which normally has nothing to do with an E3 ligase as far as we've been able to figure out. But as with other glue systems, there's a broad protein-protein interface. There's a small molecule that makes direct contact with proteins, and there is a long C-terminal extension that wraps around in blue here and makes further contact with DDB1, creating a very stable ternary complex. And that Cyclin K is the part of the molecule or part of this complex that's presented first to the E3 ligase for ubiquitination and subsequent degradation. The other interesting aspect of this particular story was that CR8 varies from roscovatine by only this 2-pyridyl group. Very similar molecules, but roscovatine is a great CDK12 inhibitor, but has absolutely no activity on degradation of Cyclin K or any other protein, whereas this addition of this 2-pyridyl group transforms the molecule into not only a CDK12 degrader, a CDK inhibitor, CDK12 inhibitor, I should say, but a degrader of Cyclin K. And the crystal study structure demonstrates that the ATP binding pocket of CDK12 is filled by the portion of the molecule that's the same between roscovatine and CR8. But this 2-pyridyl group is solvent exposed. That changes the surface of this protein that allows interaction with DDB1 in this ternary complex to form. The binding interaction on DDB1 it's a very shallow pocket. It turns out that there are a lot of molecules that will fit in this pocket and that will induce this ternary complex to form. And again, there's this very broad protein-protein interface. So at this stage, there are multiple molecular glue degrader systems, and this continues to increase. There's the CRL4-CRBN E3 ligase system, where thalidomide analogs bind to CRBN, induce interactions with IKZF1 and 3, resulting in degradation. There's a system in which indisilam and related molecules bind DCAF15, induce interaction with RBM31, and result in cell toxicity as a result. but a very similar E3 ubiquitin ligase system with COL4, and then the CR8 molecule that acts as a molecular glue with the neo-substrate receptor, not a substrate, and then the binding partner for this neo-substrate receptor, Cyclin K, is first ubiquitinated and then degraded. So we wanted to look for other systems that might be degraded, and this is another project that was spearheaded by Nikolai and was a phenomenal collaboration with Eric Fisher's lab. And this began with a molecule from Boehringer Ingelheim, and they were interested in BCL6, a very potent lymphoma oncogene translocated in many lymphomas and mutated and overexpressed in others. And they have been, that's been a long-standing project in the field to try to develop inhibitors to BCL6. They developed some very nice potent inhibitors of BCL6, but they also did Westerns and recognized that one of the inhibitors that they had developed is also a degrader. And to their credit, they made that observation, they published it, allowing us and others to investigate what the possible mechanism might be. So they targeted the interface between the BTB domain, the component of the BTB domain of BCL6 that interacts with co-repressors, N-core and B-core, that interact with this protein-protein interaction domain that is the BTB domain of BCL6. So there was no other reason for targeting that particular place in BCL6 other than it's a protein-protein interaction domain, and it's critical for the activity of BCL6. And that has been targeted by peptides, in huge small molecule screens, and they developed these small molecules that were really quite excellent inhibitors of that interaction. So we took those molecules. Eric did a proteomics experiment. You can see this beautiful degradation of BCL6 after, I should say Catherine, not Eric, did the proteomics experiment, incredibly selectively. So BCL6 goes down rapidly, no other proteins changed. And this related molecule, BI3812, is also a good inhibitor of BCL6, had no degradation activity whatsoever. So really a very, very specific molecule for degrading BCL6, and a related molecule that doesn't, which is a really handy pair to have for working out the mechanism. So we started by deletion mapping of what component of BCL6 is required for degradation. You can see that up to the first 275 amino acids of BCL6 gets nice degradation of a reporter assay, but any further deletion up to 1 to 250 completely loses degradation. And that already told us that we had a different story here, because those 25 amino acids are not where BI3802 binds. It's outside the BTB domain. So we have a molecule binding in one place and a sequence that's distal to that that's required for protein degradation. That's not what we see with glues. Still conceivable that there's some conformation change, but still gave us a clue that we might not be dealing with the molecular glue type mechanism that we've seen in the past. The other thing that Nikolai did was he looked under the microscope when he used these reporters, which are fluorescent reporters, and he recognized that when treating with BI3802, he saw these foci form very rapidly in the cell. And when he took a non-degradable form of the BCL6, added the drug, formed these foci, and he added the inhibitor that's not a degrader, he could actually dissolve the foci, and the foci would go away. And that's not something we'd seen before and puzzled us, but took note of that. Then Ho-Jung Yoon in Eric's lab wanted to make recombinant BCL6 to understand its activity and do biochemical and structural studies, and had the annoying finding that when he made BCL6 and added BI3802, it would come out of solution, which would make it harder to work with. That could be, and maybe assumed to be, a nonspecific aggregation phenomenon, but you can see in the size exclusion chromatography that after adding the drug, higher molecular weight forms were evident of BCL6. But Ho-Jung, also very persistent, continued with his studies and recognized that when he did negative stain electron microscopy, he could see these beautiful fibrils forming that of BCL6 that we could start to imagine being almost helical fibers that may be dimers of BCL6 that had polymerized. And he then solved a cryo-EM structure, and you can see that in each color here, in each band, these are homodimers of BCL6. BCL6 always acts as a homodimer. And in yellow and brown here are molecules of BI3802. And you can see that when these molecules are present, it creates a molecular glue phenomenon between dimers of BCL6. And this symmetric protein then creates an interaction with the next dimer, and it ultimately leads to polymerization, which is these beautiful helical fibers that form. That didn't explain why the protein is degraded or why it goes away. So Mikolai did the same two types of CRISPR screens that we talked about before. One for what are the genes required for degradation of BCL6. One is what are the genes required for resistance to BI3802 in a lymphoma cell. And both screens highlighted a ubiquitin ligase, CO1, that is required for its activity. With this BCL6 that Hojong had made, he could do biochemical studies, and with a TR-FRET assay, demonstrate that with BI3802, you induce proximity or interaction between CO1 and BCL6. So while it's not a molecular glue, the polymers bind CO1 with greater affinity than the unpolymerized BCL6 without the drug. And furthermore, that these two proteins, BCL6 and CO1, co-localize after treatment with BI3802. So again, after we treat with BI3802, we get these foci that form compared to DMSO-treated cells. And if we stain with for CO1 as well, you can see some punctate foci forming as well, and those foci overlap, are the same sites. So that might seem a ridiculous mechanism and highly unlikely, but we were inspired by this paper from Emmanuel Levy's lab, where they looked at symmetric proteins in E. coli and made mutants on the cell surface of those proteins, and were able to create single amino acid mutations that result in either monovalent or multivalent interactions, including polymer formation. So there may be many proteins that are on the edge of solubility or on the edge of polymerization. Of course, a single amino acid in the beta globin gene leads to sickle cell anemia through polymerization of deoxygenated hemoglobin. The same phenomenon may be true with other proteins in a cell, and a small molecule may change the surface of that protein in a way that results in polymerization or aggregation of some form. So that remains to be proven or extended beyond the BCL6 finding, but at least a possibility that there may be other proteins out there, including transcription factors, that may polymerize after addition of a small molecule. So I'll close by just noting that, of course, nothing is new under the sun, that molecular glues had been characterized before. Stuart Schreiber, of course, decades ago identified the mechanism of cyclosporin and FK506 as molecular glues that induce interactions between cyclophilin in the case of cyclosporin and calcineurin A and B, and FKBP12 in the case of FK506 and calcineurin A and B. So molecular glues have been characterized in the past. Auxins in plants through the elegant work of Ning Zhang work as molecular glues as well. So there are examples in nature, both natural products that have pharmacologic properties, as well as in plants, an entire hormone system based on molecular glue interactions, and in the case of auxins, molecular glues that result in protein degradation. I've not spent any time in this time talking about PROTACs, but again, to note that PROTACs are small molecules that bind, or molecules that have a component that bind to an E3 ligase, a component that binds to a target protein, and a linker, whereas molecular glues classically are smaller, rely on a broad protein-protein interface, and small molecules that interact with both proteins simultaneously. I'd say that that is not a dichotomy and likely a continuum, and that many of the PROTACs also rely on protein-protein interactions, and that's a phenomenon we're learning more and more about every day. And then finally, one of the reasons that we think that these are such powerful pharmacologic agents is because they are driven by occupancy rather than, by event-driven pharmacology rather than occupancy-driven pharmacology. If a enzyme is inhibited by a small molecule, you can see that the concentration of the inhibitor must exceed the concentration of that enzyme to inhibit all of that enzyme, and that in the case of event-driven pharmacology, a smaller concentration of the enzyme, of the small molecule, may induce degradation of a protein. That protein is gone in perpetuity until it's resynthesized, and so a substoichiometric concentration of the drug can have an outsized effect on its target. The mechanisms of resistance have been clarified now, I think, for myeloma and are extending to other settings, but clearly you can have loss of CRBN, loss of the E3 ligase, and that's probably the most common mechanism, either through genetic deletion or epigenetic silencing of CRBN. You can have mutations at the interface that shown here that prevent substrate binding, and at least in our in vitro models, we can demonstrate that substrate competition can result in resistance. So overexpression of a substrate that binds to the ubiquitin ligase prevents degradation of a critical substrate, neosubstrate, can lead to resistance. So these are the stories that I wanted to talk about today, all of which ended in a completely different place from where we expected, that thalidomide derivatives end up targeting an E3 ubiquitin ligase, not as an inhibitor, but as a molecular glue, that mouse cells do not respond the same as human cells due to mutations to that interface, that CR8 acts as a molecular glue, but not with a substrate receptor, but between an adapter and the substrate receptor, and that BI3802 is a molecule that's a molecular glue of a transcription factor with itself, resulting in polymerization. And an extraordinary aspect of this field has been, it's been such a collaborative field. We've been incredibly fortunate to have such great collaborators, and I think across the field, it's been a very open and collaborative field, but we've worked extremely closely with Eric, with Radek, with Catherine, with Nico Tomei and Georg and Susanna and others in his lab, with the proteomics group at the Broad Institute, and been incredibly fortunate to have such outstanding scientists in the lab, including Miklai and Jan Hojung, who's been both in Eric's lab and our lab, Jessica Gasser, Rob Seller, Adam Sperling, Quinn, Emma, and many others. So I'm happy to take any questions, and thanks again for offering me the opportunity to be here.